Hi everyone, welcome to today's lecture. Hope you guys had a nice weekend and you are feeling great. So today is a build-up of what we learned on Friday. We are going to talk about the control of microbial growth. So the objectives for today is one, to distinguish among the microbial control mechanism, such as sterilization, disinfection, antisepsis, decontamination. And then we also compare the action of microbial and microbial bistatic agent and identify factors that impact the microbial death rate. We name four types of cellular target for physical and chemical agents and the modes of action. We also identify methods and agents of physical, mechanical, and chemical control of microorganisms. And then under that, we will talk about what mechanism it fits, what mechanism you should use when you want to get rid of a certain microbial organism, such as sterilization, disinfection, abscesses, or decontamination. And what are the cellular targets that these disinfectants are targeting, whether the cell wall, cell membrane, protein, nucleic acid synthesis, or protein function. So to dive deep into it, like we learned from last time, we have to protect ourselves from microbes, and then we also have to control their growth in order for them not to over affect us. And one of such ways that we have to control them. So today the overview is two. We first talked of overview of microbial control mechanism, and then secondly we survey through different microbial control methods. So, there are four different types of microbial control mechanisms. The first one is disinfection, which is basically the destruction or removal of vegetative pathogens, but not the bacteria and the spore itself. This method is actually usually used for innate object. And then the second one is sterilization, which is the complete removal or destruction of all viable microorganisms. And this is also used again for let me try and find a pointer. This is also used again for innate mechanism. So you can see that for disinfection, it's just the removal of the pathogen, vegetative pathogen, whilst for sterilization, it's just removal of everything. And the third one is antisepsis or degermination. Basically, we are using chemicals to apply to body surfaces to destroy or inhibit the vegetative growth of pathogens. The last one is decontamination. Basically, is the mechanism of removing of most microbes. So, under microbial controls, there are three different subgroups. We have the physical agent, chemical agent, and mechanical removal methods. So, as we keep moving forward, I'll deep dive into this and describe what each one entails. So, under agents, we have two groups, which is either a acidic or a static agent. So basically what it means is that the static means to prevent the microbes from multiplying, but you don't kill them, you just halt their growth. And this agent can be bacteriostatic agent, fungostatic agent, microbial agent. And just like on a remote, you just pause your growth. Compared to the other counterpart, which is still the acidic agents, basically the aim is to kill them. So they will include germicides, microbicides, bactericides, fungicides, and virucides. And just basically just to kill the microbe. And as you can see on this graph, you can see that once we start adding the agent around the sign, when you add agents such as microbicide, which is under cedar basically to kill it, you realize that by within a short time, the quantity of microbes, which is here, the number of microbial cells goes down. It means they've been killed compared to when you add something like microstatic agent, which is a static agent. It doesn't bring the microbial quantity down, it just stops their growth. So how do you choose a method that will work for you? And we normally do this by asking ourselves some basic questions, which is, does the item in question require sterilization or is this disinfection adequate? or is the item to be reused or have to be permanently discarded? And if you also want to reuse it, can the item withstand the treatment you are going to apply? And will the agent you are trying to use penetrate to the necessary extent that you need? Will the agent also affect the environment, such as pH or temperature? And is the method costly or labor efficient? And also, is it safe for humans, animals, or plants? So when you ask this method, then you get to choose what you want to, what type of agent you want to use. So apart from that, there are factors that also affect microbial death rate and effectiveness. So these factors include two types, the type of agent you are going to use and also the microbial target. So on the type of agent, it depends on the concentration you are going to use, the time which is required, and whether the agent is acidic or static, and also the cellular mode of action in which the agent is going to affect the microbe. And then under microbial target, it depends also on the number of cells or particles which are present to clear, the organism type, and also the type of growth, whether they are in a growth phase or they are in a biofilm phase. And as you remember, it's actually difficult to get rid of stuff such as biofilm. And one example of this is seen here in this easy cleaning agent, which basically state an instruction like carefully direct nozzle under toilet rim and squeeze it and apply for 15 minutes. So as you can see, they are quantifying how much time is needed to clear off those agents, and you have to still go in and brush it and then flush. So it depends on what kind of agent you're using and a lot of factors that taking place. So, as much as we know of all of this information, we also have to know that depending on the type of microbial agent, um, they can be either resistant or less resistant. So, starting from somewhere like prions, they are more resistant to agents, and then you can have bacterial spores, which are also difficult to get rid of. We have mycobacterium. It keep coming till you get to somewhere like envelope viruses, which are actually less resistant to microbial agents and easy to get rid of. So, as much as we now know of the nature of the microbial target, the number of microbes present also taking place. So here on this graph, here is the number of viable cells of microbes present, and the time here on the x-axis is the time needed for sterilization. And as you can see, if you have few microbes, you actually need less time for sterilization compared to if you have a lot of microbes, you need more time for sterilization. So that's what you can take in context as to how much microbes are present and how much time is needed to clean. And then the next one is cellular targets. So the cellular targets are basically which part of the physical or chemical um, which part or mode of action are the antimicrobial agent targeting? Is it the physical or chemical part? So under that is the microbial antimicrobial agent targeting the cell wall, the cytoplasmic membrane, or the synthesis of nucleic acid or protein function. So to take the first one into consideration, which is the cell wall, we've discussed most of this in last class. But basically, you have an agent that is targeting the cell wall, and the mode of action basically is to damage the cell wall by blocking cell wall synthesis or digesting the cell wall or breaking down or weakening surface of the cell wall. And the whole game or the whole point is to either make the cell wall fragile or just slice through it. And as you can see in this picture, the cell was disrupted and it's sliced, and everything within the cell, bacterial cell is just coming outside. And this is more sensitive or potent in gram-positive organisms. Typically, these are agents which are used for this organism. And the second part that the agent can target is the cytoplasmic or outer membrane or viral envelope. And basically, the goal is that it targets membranes and such as biolipid membrane on the cell wall, breaking it down, disrupting it, and allowing anything else to, biolipid membrane on the cytoplasmic membrane and allowing anything to leak through. And then these are targets for bacterial cytoplasmic on outer membrane, including viral envelopes and eukaryotic cell membrane. And then their mode of action results in the loss of selective permeability. So we already know that the cell, this outer cell membrane is selectively permeable. And then when you have a disruption, it means you can rotate what goes in and come out. And then it's basically used as a surfactant. So the next one is targeting of protein synthesis and nucleic acid synthesis. So the target is actually targeting of the nucleic acid and the protein synthesis. And the mode of action, we've discussed most of this also, is to either inhibit DNA replication, the cell cannot make more copies of themselves, or inhibit RNA transcription, no more RNA transcription, and then inhibit protein translation. And also sometimes it causes mutation, which is a direct DNA damage. So let's pick the first one, which is targeting protein functions. So as we've seen, the mode of action is basically to denature the protein. So what they do is just break they basically break the protein-protein bonds. Instead of having a folded protein, you end up having an unfolded protein. And this one you can clearly see it. And you can clearly see it and in some cases, which is caused by so some of the and in some extent too, they can just denature the protein beyond repair. And some of these examples when you kind of boil an egg and then you can have it again in the liquid form. It's just irreversible damage to the protein. And sometimes it can also cause a different shape of the protein, and it can also block. Thirdly, it can also block the active site, preventing it from interacting with each other or interacting with any other substrate that can bind to it. So this is also a practice. I'll leave you guys to do it. We can also practice it in class when we meet. So now that we've finished talking about the overview of microbial mechanism, we will also talk or survey through different microbial control agents or control methods. As you can realize, this is more of a build-up of what we learned already in class. So today will be more of an overview or most of a little bit of a build-up from what we learned from last class. So now back to this figure again. So now we've understand the microbial control methods. We have to pick one specific method and go through. So what we are going to talk about is the physical agent. So under physical agent, we can have heat and radiation. And then the heat, under heat, we have dry and moist. And when we pick dry, under dry, we can have incineration or dry oven, both of which are responsible for sterilization. And under moist, we can have steam under pressure, which is for stabilization, and boiling water, hot water and pasteurization, which is for disinfection. And under radiation, we can have ionizing radiation or non-ionizing radiation. When you pick ionizing radiation, you can use things like X-ray, gamma rays, and which are all responsible for, which are all needed for sterilization. And we can have non-ionizing UV under non, which is also used for disinfection. So now, let's talk about methods of physical control. And under it, we have heat. So like I said, we have two methods. We have moisture and dry heat. And there are also other methods, which is cold, desiccation, osmotic pressure, and radiation. So as we know, some microbes are susceptible to heat. For that reason, you need both temperature, you need to consider both the temperature and length of exposure when you want to get rid of such microbes. So in some places you need higher temperature and more time. So, but what we've seen so far is that the higher the temperature, the less of a time that you need for stabilization. So for example, over here, there's a difference. This is a difference between moist and dry heat. So as you can see, under moist heat, even though it's at the same temperature as 125, 121, you need about 15 minutes to sterilize in moist heat compared to dry heat where you need about 600 minutes, even though both of them are at the same temperature. So it shows that depending on what you are going to, what methods you are going to use, whether moist heat or dry heat, it depends. You will have to determine that, and also it will also have an effect on the time that you need to sterilize everything that you have. So now let's talk about moist heat. So under moist heat, you normally have a temperature of 60 degrees to 135 degrees Celsius. And then there are conditions where you have lower temperature and shorter exposure times. And then mostly these are moist heat. you need just lower temperature and shorter exposure times compared to the dry heat method. And what it targets is that it targets the function of the protein, basically causing denaturing of the protein. And there are also three major subtypes under moist heat. The first one is boiling water. Just the goal is to just heat the water and boil it. Is for mainly for disinfection, and you need about at least 30 minutes for non-endospore or resistant organism. And the caveat is that this method is easily recontaminated because it's mostly not 100% accurate. And the second one is pasteurization, which is also responsible for disinfection, and it utilizes heat exchangers. So you have a heat of about 71.6 degrees Celsius for 15 seconds or 66 degrees for 30 minutes. The goal is that you are sterilizing it, but you also have to maintain the flavor and food value. And we've seen that it do not kill endospores and certain heat-resistant microbes such as yeast and micrococcus. And this is mainly the reason why you buy milk from the store. And even though it's been pasteurized, you leave it in the fridge for a while, it spores again because there are some microbes we couldn't take care of during pasteurization. So the third one is steam under pressure, and is responsible for sterilization. And this is more common because we've seen it in the lab. It's mostly used in autoclaves. So this one you need a pressure of about 15 psi, and you also need a heat a temperature of 121 degrees Celsius. Averagely, you need about 20 minutes to get things sterilized. And it's mostly used only on heat resistant materials. And then this is more common in research lab and in hospitals, and we've used it already in sterilizing of our media in our labs. So now let's talk of dry heat. Dry heat, you need a temperature range of around 160 degrees Celsius to several thousands, depending on how stubborn whatever you are dealing with. So at high temperatures, the dry heat oxidizes cells and burn them to ashes. So you end up just killing every single microbe that is in target. And the target is that it targets your target is to target the this method targets the protein function by denaturing the protein. And then under this, we also have two major methods for doing this. So first one is incineration, which basically means just heating things up to as much high temperature as 15,000 degrees, 1500 degrees Celsius. And basically the most common method that we normally see is like a flame or Bunsen burner, which you use a lot in our labs when we are culturing bacteria, and it's fast and effective, and it's also only for heat resistant bacteria. So if you put in something like plastic on this Bunsen burner, you end up melting it. You only need metals or like something solid like a metal that doesn't melt. So second one is hot air oven, and it's also used for sterilization. You this one is mostly you mostly find it with electric ovens, and you need a temperature of 150 to 180 degrees Celsius. And because we are using lower temperature now, it means we need more time. So you need about 2 to 4 hours for sterilization. And then you can only use this only on heat resistant materials. It's actually good for oils that steam but does not penetrate very well. So what about cold temperatures? There is not much microbes that we use cold temperatures to get rid of them. Um, because cold temperature normally slows the activities of most microbes. It doesn't just kill them; it just slows their activity. So you just target their protein function, and then you just stops or blocks the making of more protein. So they are just stuck in that vegetative state. Some microbes, such as hermeth, get killed by refrigeration or freezing, but other ones are also still active, and they just frozen in the vegetative state. Once they get the needed temperature, they will still growing back again. They will start growing back again. So now let's talk of radiation. So basically, it's just energy emission from atomic activities and then dispersed at high velocity through matter or space. So it's just basically moving of high radiation through this bacteria. And under it, we have two types. We have ionic sterilization, And then 2, we have non-ionic sterilization. So basically, the mechanism of radiation is to target nucleic acid and just incite or initiate mutations within them. In the extreme cases, you initiate DNA breaks or abnormal bonding between the nucleotides. And under this, we have subgroup of radiations. It can be gamma rays, can be X-rays, it can be ultraviolet. So with gamma rays and X-rays, they initiate DNA breakage, as we can see from here. So this is the radiation source. We have a barrier, and this is the cell, and then they initiate DNA breaks in the cell. And then compared to non-UV, non-ionizing, which is the UV rays for disinfection, um, you have basically, because UV cannot penetrate this barrier, so you also have to have the source, and then you just expose it right straight to the organism, and then it causes breaking of abnormal DNA, initiate abnormal DNA bonds and just initiate a lot of mutation within it. This is more common. The ionizing radiation is more common in supermarket. So as you can see from here, it's ionizing radiation to clean the food and prevent the growth of microbes. So now that we finished talking about the physical agents, let's now talk about the mechanical removal methods. So under mechanical removal methods, we have filtration, and under filtration, we have air, which is responsible for decontamination, and under the second one is liquids, which is sterilization. So using filtration, the target is basically nothing. It's just mechanical removal of the microbe that you are targeting. And you use air for decontamination and liquid for stabilization. And as you can see in this picture, you have a liquid or some you have a liquid and you are passing it through a filter, and within the filter there are pores within it, but the pores are so small that only the liquid can go through, and then the microbes get stuck outside of the on the filter, and it doesn't get through. And then you basically strain these through. We have filters such as this smaller one, 0.02 micrometers to about 8 micrometer pore spaces that you are filtering everything through. And then this can only be used for liquid. It's good for liquid that can't withstand heat. So if you have a liquid that will withstand heat, might not be ideal for this. So you can practice this, and we can also talk about that in class. So the third one is the chemical agent. So under chemical agent, we have gases and liquids. Under gases, we are going to need gases for both stabilization and disinfection. And under liquid, you use it on animate object and also on inanimate object. So the ones you use on animate object is responsible for antisepsis, and then the one for inanimate is responsible for disinfection and stabilization. So let's talk more of survey of antimicrobial chemicals. So we have under this, we have under the chemical agents, we have halogens. So we have halogens. These are microbicida, and this can be microbicida and can also be sporicida, responsible for targeting endospore with long enough exposure. And mostly it's more have active ingredients, which are 1/3 of all antimicrobial chemicals that we know. One prime example is the toothpaste that you have. And the groups that fall under halogens include fluoride, bromide, chloride, and iodine. And if you use toothpaste a lot, you know that it contains fluoride. It's normally used for disinfection or antisepsis, and then it basically affects protein functions. So under halogens, let's pick the first one, which is chlorine. This is one of the few that works against C. difficile, this microbe, and it's less effective if it's exposed to light, alkaline pH, or excessive organic matter. Under that, we have two subtypes. We have hypochlorite, which basically are broadly used, and Examples include the household bleach that you have to clean your bathrooms and stuff. And then we are often also used in pools and more effective at about 10%. So you realize that even in the lab, we have some bleaches where we have 10% bleach for cleaning surfaces when like we need extreme disinfection. The second group is chloramides, which are alternative to pure chlorine in water and is used for treating systems. So this one is more commonly used for treating like large water source. Like for example, treating like the water that comes to our households. So the second group that we talk about is iodine, and we have two subtypes. We have aqueous iodine and then iodophos. So under aqueous iodine, we have iodine tincture and then we have iodine tablet. So iodine tincture is just used on skin antisepsis at around 2%, and the tablets are used for disinfection of water. And then under iodophos, we have these are basically like a complex of iodine and alcohol, and they just replace free iodine solutions in medical antisepsis, but no longer in consumer products as of 2017. Um, Examples include betadine, povidone, and isodine. So under these, the next group that we talk about is also phenols. Uh, Phenols are actually the first major antimicrobial chemical being used, and it was discovered by Joseph Lister back in the days. It kills vegetative bacteria, fungi, and most viruses, but not endospores. So it's responsible for disinfection, and it's just occasionally used as antisepticism, typically because they are toxic. The phenols target the cell wall and also the cytoplasmic membrane or viral envelopes. And then the protein functions, it targets protein functions by causing denaturation of the protein. And under phenols, we have subtypes, which is phenol, the basic phenol we know, and we also have O cresol, O cresol, and also we have P cresol. So under phenols, as I mentioned on the previous slide, we have cresols, which are basically phenolic derivatives combined with soap. So you have phenol, which is combined with soap, and it's just called cresols. And we also have bisphenols, which are aerosol sprays and cleaning soaps. And then we have triclosan, which are basically disinfectant and antiseptic chemicals which are added to many products, including hand soaps. So if you use your hand soap, you realize that you have triclosans in them. And then we have the last one, which is chlorhexidine. And then they are also used in hand scrubbing, and they are used for hand scrubbing in hospitals and also surgical preparation and other medical uses, including mouthwash. So your mouthwash should have chlorhexidine in them. The last group of chemicals that we are using for disinfection is alcohols. So they come in two forms. They can either come in as ethyl alcohol or ethanol that we normally know, or isopropanol or isopropyl alcohol. Both are all effective in controlling microbial growth, and they best work as at a percentage of 70% in water. And then they are used for killing vegetative microbes and not endospores. And they are normally used for both disinfection and antisepsis. They affect the cytoplasmic membrane or viral envelope in the relation to virus. And then the what they actually do is that they target the protein function by denaturing or coagulating or causing clamp. They basically cause clampness of the protein so that they can form exact folds that they want, but they are just clamped all together. They are more effective against envelope viruses than non-envelope viruses. This is because they are targeting the viral envelope. So now let's talk much more about some of the things that contain these subgroups. So when we talk about ethyl alcohol, ethanol, these are normally used for skin and medical equipment disinfections. and they evaporate actually easily. So for that reason, they are less effective if you need things which is lasting more than 30 seconds, because you can only use it about 30 seconds and it just evaporates. Compared to isopropanol, which is isopropyl alcohol, they are more microdicidal and less expensive than ethanol. The evaporation rate also limits the efficacy compared to the same alcohol, and the vapor can adversely also affect the nervous system. Um, so you shouldn't abuse this more, because you end up inhaling it and you affect yourself too. And it's mostly more common as wet wipes, as you can see from here. So, to talk of the another subgroup of compounds which are being used, we have quaternary ammonium compounds, or quats. They are basically cationic detergents, and they affect the cytoplasmic membrane and viral envelopes that we have, and they are basically used as surfactants. They are used for disinfection mixed in a mixed environment where you have a lot of cleaning agents present. And they also can be used as antisepsis, basically preserved for ophthalmic solution and cosmetic. They are mostly found in the form of microbicidal to most vegetative organisms, but not certain pathogens. So an example include a hepatitis virus. They are basically like a low-level disinfectants in clinical applications. So you find most of these uh, either in the clinics or maybe in the drugstore. This one particularly. So we have soaps, which we already have. These are basically alkaline compounds mixed with fatty acid, um, with sodium or potassium salt. They are they are ionic detergents which are less effective than cationics, and they are used along they are basically used along mechanical removal of microbes. So it just means if you have them, you have to mechanically move your hands or whichever surface, scrub it or move your hands just to clean, because without it they just don't clean. You have to add some mechanics to it, and then they affect the cytoplasmic membrane or viral envelopes, and then they are used in the form of either antisepsis or disinfection, and they are actually weak microbicides, um and they've been in some times where we have pseudomonas growing abundantly in soap dishes, even though soap is present, but it doesn't target these microbe pseudomonas. The next on our list is hydrogen peroxide, which is a bactericidal, a virucidal, fungicidal, and then sporocidal at high concentrations. Um, they are basically considered a reactive oxygen species. So they affect DNA, RNA damage, and then protein function. So under it, we can use it as antisepsis. Basically, when you have some wound or skin, um you have a wound on your skin, you can use it for cleansing. We can also use it for bed sore cares and also mouthwashes. We have them as mouthwashes, and we can also have them as disinfection. And we can also use them for disinfection, which includes soft contacts, surgical implant, plastic equipment, and we can also use them for sterilization in the form of vaporized hydrogen hydroxide, and we also have them in small industrial items, and also we also use them in small industrial sterilizing small industrial items and also larger enclosed spaces. So what are the negative effects of antimicrobial chemicals? As much as they have good part, they also have the bad part. Um, The bad part is one example of the bad part is triclosan. So it's just basically excreted in the urine of about 75% of Americans. So they are detectable levels in groundwater sources. So even though you are using them and you have this even though we use chemical microbial agent, we have some that is more after we've taken them or we use them again ourselves, they get into our urine, we end up peeing it. and we find them in the groundwater. So back to what I was saying, they degrade into dioxide-like compounds when exposed to sunlight at the risk of the environment. They promote resistance to triclosan and other antibiotics in bacteria. So as much as you are using these antimicrobial agents, if you use things like agents such as triclosans, they get absorbed in your bloodstream. You end up peeing on the ground or whatever, and then it end up being in the underground water sources, which now comes back to us as toxins. So please, you can practice this. When we meet in person, we can talk more about it. Also, remember to practice the control of microbial growth and prevention worksheet. This is all we have to talk about today. Um, Next week Friday, I think see you next week Friday. I think next week Friday is Good Friday, so then it should be the next week Monday. So try and turn in all your assignment on time, and remember we have TA hours always. We have TA hours always on Mondays and Wednesdays, the same time as the Friday lecture time, and in the same room or on Zoom. See you and have a nice day. Bye.